Okay, so urinary system. As you know, we have two kidneys, two ureters, one bladder, and one urethra. Uh, Kidneys um, have a very important function of filtering blood, but not only filtering blood, also reabsorbing a couple of things from the urine. Because the mechanism of filtering is very primitive. Basically, anything which is less than 60 kilodaltons is going to be filtered. So if you're smaller than 60 kilodaltons, you will pass through the barrier and you will be excreted. Water is smaller than that. Glucose is smaller than that. Vitamins, amino acids, nutrients, all of those things are smaller. So that's why kidneys, in addition to filtering the blood, they also have to reabsorb the nutrients. You wouldn't want to pee your vitamins out, right? Control water ion salt balance, acid base balance, excrete metabolic waste, of course. But also kidney is a endocrine hormone, and that's a question in the self-control test. That literally that's the statement that kidneys are endocrine hormone, and that's correct. They make a couple of hormones. One of them is a calcitriol. Do you guys know? Does anybody know what is calcitriol?
It's activated vitamin D3.
Amazing. It's the activated vitamin D3, exactly. So, as you know, we get vitamin D through the sun, right? So, we are supposed to be exposed to sun so we can get some vitamin D to it. But it's not active, it needs to be converted to its active form. And it is done by the kidneys. So, kidneys will produce calcitriol. So, that's why whenever you actually have a problem with bones or something, they don't only prescribe you calcium, they also prescribe you vitamin D as well, because your kidneys will convert it to the active form, and active form is calcitriol. You have 3 calciums available per molecule, so that's a lot. Kidneys are extremely vascular organs. They actually receive 25% of the cardiac output. That's a lot. Okay, so for 2 kidneys, that's a lot. And finally, urine will contain water, electrolytes, and waste products such as urea, uric acid, and creatinine. In the oral, they could ask you this question: which of the waste products is used to monitor the function of the kidney? Which one would you use to monitor the function of the kidney? Anybody knows? It's actually creatinine. Okay, so as a doctor, when you order lab, you ask for the creatinine value in the urine and in the blood to see if your kidneys are doing fine. Because if there's more creatinine in the blood and less of it in the urine, then your kidneys are failing, right? So they're not doing a very good job of excreting it. So creatinine is used to monitor the function of the kidneys. Anybody knows what is creatinine? It's a
Intermediate feeling?
sorry.
Intermediate feeling?
Mm.
It's stored
You're,
in muscles as a form of energy.
it is. It's actually, it's kind of like a byproduct of the muscle. Yes. So muscles, they use some sort of things for energy, like a building blocks for energy. And creatinine is the byproduct of that. So basically, in a way, like for example, if you compare a normal person with the bodybuilder, who will have more creatinine in their blood? Of course, bodybuilders, because they're actively using their muscles, right? So generally, creatinine is used to see how well your muscles are doing. Like, are they are they working? Because muscles have a very normal thing where they break down and then they build up. You go to gym, you tear your muscles apart, but then you have to have a rest day so they can build up, right? So they can go back to their normal way as well. So that's why creatinine is just like a breakdown product. We have said that endocrine is that the kidney is the endocrine organ. So of course it makes the calcitriol, which is the activated form of the vitamin D3. For sure, you remember that it makes renin. Renin belongs to which kind of system? How would you call the system?
RAS.
Aldostrones.
Right, so there are two A. What is the second A?
Angiotensin active.
And A?
Renin angiotensin activated.
Yeah, I know that they use aldosterone. Yes, yes. You usually use aldosterone. So yeah, there is your confusion with the enzyme because there is the enzyme that is different there, but generally the system is called RAS. RAS is renin angiotensin aldosterone system. Renin is basically used to convert angiotensinogen to angiotensin 1. And as you know, angiotensin 1 is needed to release aldosterone. So what is the end product of the RAS system? Basically, what's this RAS system is for?
Reabsorption of water.
Water and salt.
So what's the end product? What's the result?
Aldosterone.
The blood pressure goes up.
High blood pressure.
Blood pressure, very good. So the RAS system is needed to increase your blood pressure. How exactly? We have talked about it last self-control test that aldosterone it has a one job. It's to reabsorb sodium. If you reabsorb sodium from the kidneys, water will always follow. And all of this reabsorption is in the blood, so into the blood vessel. So this water goes to the blood vessel, increasing the blood volume. And in physio, you have learned that if I increase the blood volume, the blood pressure goes up. So RAS system is to increase the blood pressure, overall blood pressure. And another hormone that kidneys are making is erythropoietin. And we talked about it last semester. Do you remember? What does erythropoietin do?
Formation of red
Stimulate the
blood
bone
cells.
marrow.
So it stimulates erythropoiesis, production of the red blood cells. What acts as a stimulus for its secretion?
Low oxygen.
Amazing, which in medicine we call hypoxia. Very good. So kidneys are constantly filtering blood. As they are filtering blood, they can detect low oxygen level or hypoxia. They do not know how many red blood cells there are, so they think maybe there aren't enough red blood cells to carry the oxygen. So they secrete erythropoietin. Erythropoietin goes to the red bone marrow and stimulates production of the red blood cells. Capsule of the kidneys. Kidneys, as any organ, has a capsule, but its capsule is very special. The outer layer is normal. It consists of the fibroblasts and connective tissue like collagen. But the inner layer is very special because it possesses myofibroblasts. Could you tell me which other organ has myofibroblast in the capsule?
Spleen.
Very good. It's going to be spleen. So there are two organs, kidneys and spleen. Within their capsule, you'll find myofibroblast. In the spleen, as you remember, these myofibroblasts had one job. They were contracting, and as they were contracting, they were squeezing the remnant blood from the spleen whenever it is needed. In the kidneys, myofibroblasts do not have this kind of function. Instead, they also contract, but they contract because they need to resist the volume and the pressure changes. Your kidneys sometimes receive more blood than usual. And as you know, they receive 25% of the cardiac output. That's a lot. So what prevents kidneys from simply bursting because there's so much blood? Myofibroblasts. They have this contractile ability where they kind of contract and resist this volume and pressure changes. Okay, hilum, you remember, right? So that's the entrance where blood vessels enter and leave. Structurally and functionally, the kidneys can be divided into two. There are two things in the kidney. That's the nephron and collecting system. In simple English, collecting system is collecting urine. But what is nephron doing? It's producing urine. So how exactly? By filtering and excreting unnecessary substances. So kidneys structurally, very important, and functionally are consisting of two, nephron and collecting system. Without going too much into the details, nephron itself consists of, here we have a renal corpuscle. As you can see, inside of the renal corpuscle, there's like a blood vessel. this blood vessel is being filtrated, and whatever is unneeded or whatever is less than 60 kilodaltons is being pushed into the space which is outside of this blood vessel. The space here is called urinary or Bowman's space, right? Then this urine flows to the proximal convoluted tubule. From there, it flows to the loop of Henle, and then to the distal convoluted tubule. That's your nephron. That's what nephron consists of. Renal corpuscle, proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Along this way, you can modify your urine. You can absorb things or add things. You can still produce the urine. From the distal convoluted tubule, the urine flows into the collecting duct. And in the collecting duct, you cannot modify the urine. That's it. That's the end product. So you're just collecting the urine there, right? So as a quick, quick overview, this area here, that's the nephron. It's the filtering, excreting system, which is producing the urine. And this part is the collecting system. Here we're talking about general overview of the kidney, which I will explain on the picture. So here is your normal kidney. So disregard, that's not the nephron, it's something else. So this is general view of the kidney. And you can see that there are two tissues to it, right? So there's like this outer tissue that looks as one thing. And then there is the inner tissue here, which looks like a completely different thing. They do not look the same. So they have a different function. If they have a different function, do they have the same embryologic origin?
No.
No. So that's why it's a very famous and favorite question by Professor Antal. Where does the kidney come from? And it's a trap question because you cannot answer this question. You have to ask which part of the kidney are we talking about specifically, because don't forget, kidney has two things. It has nephron and collecting system. So you have to ask which part are we talking about, the nephron part, the filtering, excreting part, or the collecting part of the kidney. In embryo, we will learn that they do not have the same origin. They come from different things. Without going, again, too much details, if you take a look, we can just say that the outer part, this is the cortex. And this is the medulla. And this is your collecting system. It's like big, huge collecting system right here, the visible one. As you can see, the medulla of the kidney is arranged into this triangular looking something, okay? So triangular looking structures. And they are called pyramids. So sometimes they're called renal pyramids. How many there are, it depends. It could be 10 or 18. So every person has them different. So I repeat, medulla is arranged into this pyramidal structure, triangular structure. So you call it renal pyramid. Overlying is the cortex. So how do you call this structure, this one renal pyramid with the overlying cortex? This is known as the renal lobe. Okay, so the structure that they're pointing to you, this is a renal lobe. It's a picture from the internet, so I took it there, but it's a bit wrong. So renal lobe, this is renal lobe right here. Now, okay, could you please, you remember this, right? So renal corpuscle, proximal convoluted, loop of Henle, distal convoluted, and collecting duct. I want to reapply it to this picture. So where are these structures on these pictures, for example, right? So renal corpuscle is somewhere in the cortex. That's actually the major difference between cortex and medulla, that you will never find renal corpuscles in the medulla. All of them are present in the cortex. What is going from the renal corpuscle? What is coming out from the renal corpuscle?
Coming out?
Coming out from it, yes.
Blood.
Proximal collecting.
Filter here, it's the urine already flows this way. So urine is coming out from the renal corpuscle, but into where?
To the proximal.
Proximal convoluted. So if this is my dot, I'm going to draw it a bit big. as one, but you can imagine that there are many, right? So this is my renal corpuscle. From my renal corpuscle, I have proximal convoluted tubule like this. What is coming out from the proximal convoluted tubule? What is next?
Distal convoluted tubule.
Loop of Henle. And this loop of Henle will be dipping down. It goes like this. This is my loop of Henle like this, right? So this is loop of Henle. From the loop of Henle is
The
Distal.
distal
distal
convoluted.
convoluted tubule right here. That's the distal convoluted tubule right here. From the distal convoluted tubule, I have
Collecting.
collecting duct, which is also dipping down. So a question to you. What can you find in the medulla? Basically, what does this renal pyramid consist of? What is inside of it?
Loop of Henle and
Loop of Henle and collecting duct. Agree with me that this collecting duct will be called medullary collecting duct because it is within the medulla. And this part of the collecting duct will be called cortical because it's in the cortex, right? So you can see it's one collecting duct, but it is into two different areas. So if we say it a bit better, what is within the medulla? Within the medulla, we can see loop of Henle, a part of it, and medullary collecting duct. At the bottom of the medullary collecting duct, you can see the area here, which is called papilla. Papilla is this tip of your renal pyramid, of this triangular area. And in the papilla, you can see papillary collecting duct. Okay, so here we have papillary collecting duct. So it kind of is continuation of the medullary collecting duct. Papillary collecting duct has another name. It's also called collecting duct of Bellini. Okay, so in anatomy, it could be important. Italian dude, right? So collecting duct of Bellini. This is the papillary collecting duct. Papillary collecting duct opens up into this area. Do you see there's like a gap here? They didn't point out for you what is it, but in anatomy, it has a name. It's called area cribrosa. Okay, I wrote it somewhere else, but for now, it's called area cribrosa. So papillary duct opens up into the area cribrosa. Area cribrosa opens up into the minor calyx. You can see that there are several of them. Minor calyx or minor calyces open up into the major calyxes. You can see them here. There are about typically 2 to 3 of them, not more, just 2 to 3 major calyces. And major calyces open up into the renal pelvis. So it's a huge structure here, for example, here, right? So that's the renal pelvis. Renal pelvis continues as the ureter. And then from the ureter, you go to the bladder. So can we do the pathway again? Because it's quite important before we move on to the next part. So you start with the renal corpuscle. What is next?
Proximal
Proximal convoluted, which is located in the
cortex.
cortex. This
Collecting duct.
Loop of Henle.
loop of Henle. Still forgetting. So loop of Henle. This one,
Distal.
distal convoluted tube. So you can see it kind of dips down and then up again to the cortex. This is distal convoluted tubule. Then cortical collecting duct. Then medullary collecting duct.
Papillary.
Papillary collecting duct.
minor calyx.
Before it, there is this area, which we call area cribrosa. Okay, so papillary duct opens up into this, I want to say bucket, because it is kind of in a way a bucket that is just collecting stuff. So this is the area cribrosa, this part. And then from the area cribrosa to the minor callus, then to the major, and then to the renal pelvis, then to the ureter. Is it clear so far?
Yeah.
Again, renal lobe. It's very important. It's a pyramid, renal medulla or renal pyramid with the overlying cortex. Because now we have to answer the question, what is renal lobule? There is a renal lobe and there's a renal lobule. So renal lobe, I repeat, is just renal pyramid with the overlying cortex. That's it. So again, the structure is quite similar. You can see that here's the pyramid. This is the cortex and this is the medulla. you can see that there is a part of medulla which kind of protrudes into the cortex, right? And we call it medullary ray. So medullary ray, I repeat, is in the cortex. It's inside of the cortex, but the structures that it has belong to the medulla. Anybody could guess which parts is the medullary ray? What is inside of it?
Loop of Henle.
Amazing, amazing. Loop of Henle and
Collecting duct.
the
collecting duct. Amazing. But which collecting duct specifically?
Medullary
The
collecting
medullary.
duct.
Cortical, because it's in the cortex. So medullary ray has cortical collecting duct. Because again, guys, medullary ray is in the cortex. Collecting duct should be in the medulla, but it is in the cortex, so you call it cortical collecting duct. So basically, which part is medullary ray? This part, this area right here. Do you see? So this part, this is your medullary ray, where you see a bit of a loop of Henle together with the cortical collecting duct. Do you see this part? This is still cortical collecting duct. So this is your medullary ray. Medullary ray has another name. In Latin, it is called pars radiata because it's standing straight, it's kind of radiating forward. So this is a pars radiata. So medullary ray is a pars radiata. Could you tell me what is outside of medullary ray? What could you find here?
Bowman's capsule.
The corpuscle.
We haven't
Glomerulus.
mentioned Bowman's capsule yet, so I would agree renal corpuscle. So these dots that you can see here, these red dots, they are renal corpuscles. What else?
Proximal.
And tubules, such as
Proximal.
proximal convoluted and distal convoluted. This is why this area in Latin is known as the pars convoluta. So surrounding pars radiata, you have pars convoluta. So if I go back, this is my pars radiata. Everything else is pars convoluta. Everything else is pars convoluta, which is outside of my medullary ray. What is there? What we just said, renal corpuscle, proximal convoluted, distal convoluted is there. So what is renal lobule? Renal lobule is this. So could you tell me what is in the center of the renal lobule?
Medullary rays.
Medullary ray, yes, medullary ray is in the center of the renal lobule. What is inside of the medullary ray?
The
Loop of Henle.
collecting
And the
Collecting
collecting
duct.
cortical collecting duct.
Cortical collecting duct. What is outside? What is in the periphery of the renal lobule?
pars convoluta.
Renal corpus.
Convoluta, pars convoluta, which consists of
The renal corpuscle.
Proximal and
distal and
distal.
proximal.
proximal and distal convoluted tubules. Is it clear?
Mhm.
Okay, so that's, I guess, the most important thing there.
Yeah.
Ah.
The medullary rays are like infiltrating the medulla. Like they go inside the medulla.
It goes inside of the medulla, yes. It's like basically because inside of it is the loop of Henle. And you see when we draw it, loop of Henle begins in the cortex, but it is in the medulla. So in a way, it is like a part of the medulla, which is in the cortex. So that's why they called it medullary ray. It's just this little part of the loop of Henle, which is in the cortex, because it should be in the medulla. It should be deepened down, but you see sometimes it's there. Not sometimes, all the time it's there. So again, I will be adding more details. So just don't rush, and you will see there's so many details to add on. It's insane. But eventually, if you listen carefully, at the end of the class, you're either completely confused or everything is clear as day. Okay, so it's one of the two. There is no middle. So half of the class is typically like, yeah, that was so understandable, and half of the class were like, what the hell was that? Okay, so they don't understand anything. So that's why recording is there, so you can again and again do it, because it's the most important thing. Okay, so again, going back, we have renal lobe. This is the renal pyramid with the overlying cortex. And in the cortex, we have renal lobules, inside of which medullary ray is the center. And this cortical area is the periphery. In the medullary ray, we have loop of Henle and cortical collecting duct. In the pars convoluta, we have everything else, which is renal corpuscle, proximal, and distal convoluted tubules. So now we come for this part again. So could you tell me how would you call all of this? So this area, how do you call it?
Nephron.
Pars
Nephron. Very
convoluta.
good. This is nephron. So this is your renal corpuscle together with proximal convoluted tube, loop of Henle, and then distal convoluted tube. This is called nephron. And then this is the collecting system. How do you call it at the bottom here?
Papillary.
Papillary duct. Yeah, so here it will become papillary duct. We have said that kidney is going to be filtering blood. And the filtering mechanism, as we have discussed, is very primitive. So if you're smaller than 60 kilodaltons, you're going to be here. You're going to be within these tubules. So supposedly, for example, I have, I don't know, glucose that is in my loop of Henle. I don't want to pee out the glucose because maybe somebody else in my body needs it more. So I want to reabsorb that glucose, for example, from the loop of Henle. Where would I reabsorb that glucose into?
Renal corpuscle.
Renal corpuscle is here. It's too far away for me to go there. So something next to the loop of Henle should be there, so you can easily reabsorb things into it. You can easily put it there.
Capillaries.
Blood vessel. Very good. Just a blood vessel. So right here next to my loop of Henle, I'm going to have a blood vessel, which will be accepting anything which is not meant to be excreted. So for example, glucose or water. So how do you call a blood vessel which accepts this reabsorbed things from the loop of Henle specifically? You call it vasa recta. Okay, so if I am reabsorbing from the loop of Henle, for example, glucose, I don't want to pee it out, I put this glucose into the vasa recta. Can I reabsorb things from my proximal and distal convoluted tubules? Of course, because again, nephron, you're still making the urine, you're still modifying things, want to add some things, want to reabsorb some things, and etc. Can distal convoluted tubule reabsorb into the vasa recta? No, it's a bit too far away. So that's why convoluted tubules have another capillaries that are adjacent right next to them. And they're a bit tortuous like that, just like them. These capillaries are called peritubular capillary network, peritubular capillaries. So if I have, again, the same glucose that is in the proximal convoluted tubule, but I don't want to pee it out, where would I put this glucose into? Into the peritubular capillary network. Water is in the distal convoluted tubule. I don't want to pee it out. Where would I put it? Peritubular capillary network. Sodium, not sodium, bad example. Glucose, still glucose, is in the loop of Henle. Want to reabsorb it. Where would I put it? Vasa recta. So remember, the two vessels that will be used for reabsorption, where you would put things into, are peritubular capillaries and vasa recta. Peritubular capillaries are specifically for the convoluted tubules. Vasa recta is specifically for the loop of Henle. Is it clear? Another way. So there's one area here, there's one area here. In other way, how could you say where are vasa recta? In which part of the kidney are they?
Medulla.
Very good. In the medulla. They're in the medulla. Where are the peritubular capillaries? In the cortex. Could there be vasa recta in the cortex?
I feel like that's a trick question.
Sorry?
I feel like that's a trick question.
That's what I'm asking. So could there be vasa recta in the cortex? So yes or no? Try to think
Yeah.
before saying.
I'm gonna say yes, but it shouldn't be though.
If I'm asking, that means it's yes, okay? It's always yes. Okay, so it's always yes if I'm asking. So of course, yes, there will be vasa recta in the cortex, but where exactly?
Maybe in the radiata zone.
Amazing. In the medullary rays. Don't forget, medullary rays contains loop of Henle. If there is loop of Henle, there is vasa recta. So medullary ray is in the cortex, but it contains loop of Henle. So there will be vasa recta in the cortex as well, but specifically in the medullary rays. A very unfair question that they could ask you has something to do with this part, counter current exchange system. Basically, you learn more details about it in physiology next semester, because half of the physio semester is kidney, and our half is endocrine system. So you spend so much time on kidneys. Basically, without going too much details, if they ask you this question in the oral, the way urine flows in the loop of Henle, specifically in this part, it flows downwards. So the way it goes downwards. The way blood is in vasa recta, it goes upwards. You will see it, what I mean in the actual part. So do you see they're going in opposite direction? That creates counter current. So they're going in opposite direction. Because they're going in opposite direction, specifically in this area, it's the easiest way to exchange things, because this current creates a gradient. So you can easily diffuse back and forth. Okay, so that's why if they ask you how exactly reabsorption happens, why things go to vasa recta, because of the counter current exchange system, because the way fluids are flowing in those two areas is opposite direction. The urine in the loop of Henle is going downwards here. The vasa recta blood is going upwards. It creates a current. This current is needed for exchange. Okay. This is not important, it's just like a very weird question in the oral that could be asked. Basically, as you know, we have renal lobes, right? So there you go, renal lobe, renal pyramid with the overlying cortex. My question to you is kidney lobulated?
No.
No.
No. So it's, even though it has lobes, even though you see the functional part is the lobe, if you were lucky enough to see a kidney, it's smooth. It's smooth. It has no lobar appearance. It's very smooth. So only in embryos do you see this lobar appearance. Do you see that there are some gaps there that create this lobar? Only in embryo, because you know, kidneys are very important in embryo. They do majority of the filtering. So of course it's you, when you make the kidneys, you need to make as much of them as possible. So they actually have this lobular appearance like this. But by the time you become adult, the kidneys become much smoother. Okay, so they become super, super smooth. So you don't see this lobulated appearance. Everything else, I don't think it's important. Back to the medullary ray, right? So you can see that we're in the cortex. There is something here which is quite straight. This is medullary ray. What is another name for it? Pars.
Pars radiata.
So it's also known as the pars radiata. What is outside of it like this? What is here?
Pars
Convoluta.
convoluta.
Yes, as it says, so this is pars convoluta. Instead of the pars convoluta, you can see something. What is this?
The corpuscle?
Yes, these are the renal corpuscles, and the rest of them are tubules. So these are your proximal and distal convoluted tubules. So how can I know that this is a border of the renal lobule? Because you remember we have discussed, right? So this entire structure is a lobule with the medullary ray in the center. So this is a renal lobule. So how do I know? Blood vessel will help me. So always at the border, there will be a blood vessel. How would you call this blood vessel?
Interlobular.
Amazing. Interlobular. How would you call a blood vessel here?
Intralobular?
Interlobar. Because it's a lobe right? So the vessel between the lobes will be interlobar. So here, these vessels, they're interlobar. But where the lobules? They're in the cortex, they're here with the medullary rays. So that's why these little tiny vessels right here, they're interlobular. So remember the difference, there's a lobar and lobular. So it's not the same thing, that's what I'm trying to tell you. So this is a lobule, it's not a lobe, it's a lobule. So the vessel here is interlobular. So that's why it's very easy. All you have to do is just find this vessel, this interlobular, typically vein. Majority of the time it is actually vein. Draw imaginary line, and that's the border of the renal lobule. Okay, so the same situation, medullary ray, right? So pars radiata, and you can see here pars convoluta with the renal corpuscles and the tubules. Find the vessel. Okay, so this must be my interlobular vein. There's another vessel right here, right? So interlobular vein as well. Draw imaginary line like this. There's one here, there's one there. Whatever you have between these two lines, that's your renal lobule. If that's the renal lobule, the vessels here are called interlobular. This is also a medullary ray, but from a different section, because here this is a longitudinal section. It's almost like coronal, okay? So it's longitudinal section. This one is a cross section of the medullary ray. So you can see here some lots and lots of ducts. So don't forget, what is inside of the medullary ray?
Loop of Henle and collecting and cortical collecting duct.
Very good. So here we should have loop of Henle and cortical collecting duct. So now let's talk about renal corpuscle to be a bit more details, right? So we have said that there is renal corpuscle, it's filtrating, but what does it actually consist of? Renal corpuscle has another name that we have encountered before. Do you guys remember?
Bowman's capsule.
In spleen, we saw it. I told you not to call it like this. Because Malpighian corpuscle, very good. Okay, so in spleen, we said there is white pulp. White pulp has another name, it's called Malpighian corpuscle, but I told you not to call it because renal corpuscle is the definite Malpighian, right? So Malpighian corpuscle, that's another name for it. What does Malpighian corpuscle consist of? Inside of it, you see glomerulus. Okay, so that's in the center of it, glomerulus. What is glomerulus? It's like a knot. I swear, if you use this word in the oral, they will be ecstatic, okay? Because everybody says tuft because it's in the book and in the lecture, but nobody understands what the tuft means. Knot is much better said, so because that's what it is. So glomerulus is a knot. Everybody knows what knot is, right? So you're tying a knot, right? So knot. It is a knot of what? It is a knot of blood vessels. To be more specific, fenestrated capillaries. So this glomerulus is a knot of fenestrated capillaries. How big are the fenestra? Well, about 60 kilodaltons. But every semester they change the number, and I gave up trying to memorize, so that's why you have homework to look at your lectures, okay, or handout. They should write a number there. How big is this fenestra? And you're gonna go with that fenestra. I'm just sticking to the book. In the book it says approximately 60, sometimes 70 kilodaltons. So you're gonna go with the lectures, whatever lecture says. So they have some kind of holes here, fenestra, and these fenestra are about 60 kilodaltons. Glomerulus, which is a knot of fenestrated capillaries, is surrounded by a capsule, which is called Bowman's capsule. Bowman's capsule has two layers, visceral and parietal, as you see anywhere. But visceral cells of the Bowman's capsule have a very interesting appearance. As you can see, they have lots of these food processes coming out. That's why the visceral layer of the Bowman's capsule are also known as the podocytes. Podo is food, like podiatrist is a food doctor, right? So podocytes. So these cells are podocytes. You can see they're on the top of your glomerulus. They are visceral layer of the Bowman's capsule. Parietal consists of simple squamous cells, nothing interesting. Between podocytes and parietal cells of the Bowman's capsule, you can see that there is a space here, and this space is called Bowman's space, but another name for it, it is also known as the urinary space, because whatever is being filtrated will go to this urinary space. It will be squeezed out of the blood vessel into the urinary space. What is feeding this glomerulus? As you can see, there are two vessels here, afferent arteriole and efferent arteriole. The area where they're located, this is known as the vascular pole. Like, for example, North Pole, South Pole, right, of the Earth, we have a we have a renal corpuscle. It has a vascular pole, which is having the blood vessels. And at the bottom of it, it has a urinary pole. So this is the opposite end where the urine will be kind of accumulating, right? So at the vascular pole, you can see that there are two blood vessels, afferent and efferent. Guess which one is the feeding one? The one that feeds the glomerulus blood?
Afferent.
Afferent.
Of course. So afferent is going in. So the blood from afferent is coming in into the glomerulus. It gets filtrated and then it's coming out from the efferent arteriole. At the bottom of the urinary pole, a tubule begins, which is called proximal convoluted tubule, right? So you can see that's where it starts. So whatever is being filtrated goes to the urinary space, and then from the urinary space to the urinary pole, and then from there to the proximal convoluted tubule. For later purposes today, remember that at the vascular pole, there's also a tubule present, and it is distal convoluted tubule. So at the urinary pole, there's proximal convoluted, and it begins there, it starts. At the vascular pole, there is distal convoluted tubule, and it doesn't start there. It's just very near it. Do you see it here? So that's your distal convoluted tubule, and it's kind of approaching the vascular pole. You can see it here, right? So it's the distal convoluted tubule. That's the proximal. Okay, very unfair question, but what makes things to leave blood vessel and go into the urinary space? Why would they do that?
Hydrostatic pressure is different.
It's the difference in pressures. As you know, in physiology, you have learned that we have two, oncotic and hydrostatic. So tell me, where is the oncotic pressure higher? In the glomerulus or in the urinary space? And why?
In the
The
glomerulus.
glomerulus.
The glomerulus. Oncotic pressure is much higher in the glomerulus. Why?
You have more proteins cannot filtrate.
Exactly. Proteins cannot be filtered. The tiniest protein there is albumin, and it is bigger than the pore itself. So it cannot be filtered. So the reason why oncotic pressure is higher in the glomerulus is because proteins cannot leave. They stay where they are. Hydrostatic pressure will be the opposite. It will be higher on the outside. So it will be literally dragging things out. You're going to see this. It's going to be attracting things to come out, to join the dark side. Okay, so join me to come this way and stay in this area. So of course it is just pressure differences. It should be enough for the oral. They shouldn't go details because again, you haven't learned physiology of the kidney yet. You're going to do it next semester. So just remember that there is a difference in pressures. Now we come to the details. So you remember that this is the renal corpuscle with the glomerulus and Bowman's capsule. This is proximal convoluted tubule. This is loop of Henle, right? Distal convoluted tubule, cortical collecting duct, medullary, and etc. Loop of Henle actually consists of 4 parts. There are 4 things to it. This part is called thick descending. This is thin descending, then thin ascending, and then thick ascending. So loop of Henle has 4 components, right? Thick components and thin components. Thick components, however, have another name. Thick descending is located very close to the proximal convoluted tubule. That's why thick descending is also known as the proximal straight tubule. So proximal convoluted continues as the proximal straight, right? Thick ascending is very close to the distal convoluted tubule. So how would you call it?
Distal straight.
Distal straight. So can we do the pathway again? So renal corpuscle, then
Proximal
convoluted,
convoluted.
then
Proximal straight.
proximal straight. Yes, you should be saying proximal straight, then
Thin descending.
thin descending,
Thin ascending.
thin ascending,
And distal straight tubule.
distal straight, and then
Distal convoluted.
And
distal
distal
convoluted.
convoluted.
Now a very difficult question. How do you call this?
A nephron.
Cortex.
Nephron is all of it.
The
How would you
cortex.
call this part specifically?
Medullary ray.
Amazing. Medullary ray. So now you're going to change your answer. What is exactly in the medullary ray?
Thick. And
Proximal straight part of the loop of Henle, distal straight part of the loop of Henle, and cortical collecting duct. This is the medullary ray. Is it clear? Okay. So that's what we talked about. Just read at home. There's some additional thing, but yeah. This is an overview. So you start with the glomerulus from where you go. So it's very useful for you. Just do it at home as well. Now, types of nephron. We have said that this is the nephron, this entire thing. All of this is your nephron. And there are 2 types of nephron. Superficial cortical and juxtamedullary. In the book, they also put intermediate, but so far in the lectures, they disregard it, so it's not there. So in the lectures, they always, for the past 2 years, they've been only putting these ones, so superficial cortical and juxtamedullary nephron. What's the difference? Well, if you take a look at it, this is what you can see here. So this one is superficial cortical nephron. So its renal corpuscles are a bit more to the cortex, right? So they're a bit upper in the cortex. Juxtamedullary, and this picture is also not giving you justice, but it should be a bit this way actually. So renal corpuscles are right at the border between the cortex and medulla. That's why you call it juxtamedullary nephron. My question to you is, which ones will have longer loops of Henle? Superficial cortical nephrons or juxtamedullary?
superficial.
Juxtamedullary, because they have to put their loop of Henle through the entire thickness of medulla. So actually, whatever loops of Henle you see in the medulla, they will belong to juxtamedullary. So you will see that they have there in the medulla proximal straight, right? Thin components, and then distal straight. So if you see it, that means that this part belongs to the juxtamedullary. How do you call this part?
Medullary ray.
Medullary ray, like this. Sorry, I forgot the collecting duct. Okay, so this is called the medullary ray. So in a way, medullary ray belongs to which nephron?
Superficial.
The
Superficial cortical. It's not belonging to the juxtamedullary. It's just this part which is a bit upwards. Yeah.
I don't get why.
just the way it was made. There is, in physiology, we learned that juxtamedullary have a much nicer glomerular filtration rate, but you will learn it in physiology. Basically, the juxtamedullary, they filtrate much better because they're through the entire thickness of the kidney, so more area to filtrate, right? So if I have a longer loop of Henle, I pass this urine through a much longer way, so I can modify it better. I have more time to absorb things, reabsorb things, etc. Superficial cortical are also useful, but they're just a bit shorter. So they are there through embryological development.
So Yuri, I have a question. The juxtamedullary nephron, is it a part of the medullary ray?
No, that's what we're trying to understand. So medullary ray are the thick components of the loop of Henle that belongs to the superficial cortical. Because juxtamedullary, I told you, its glomerulus, its renal corpuscle is not here. It's actually here. It's right at the border. So medullary ray is here. Their loop of Henle begins here. It's already in the medulla. So no, medullary ray is the part of the loop of Henle that belongs to the superficial cortical. Okay, filtration apparatus of the kidney, we know everything. So that's your glomerulus, has endothelial cells. In the book, it says 60 to 70 kilodaltons or nanometers. Last year, in the lectures, they wrote 70 to 90 kilodaltons. Okay, so you're gonna check this year what they wrote. Hopefully it's the same. Okay, but yeah. Basement membrane, which is right under the endothelium of the fenestrated capillary. It's very thick here. It's quite thick. Why? Because it's not only produced by the endothelial cells, but also by the podocytes. A Histo 1 question for you. Basement membrane, does it belong to the epithelial tissue or to the connective tissue, and why?
The connective tissue.
To the epithelial tissue, but why? If it belongs to the connective tissue, which cell will make it?
The
Fibroblast.
Fibroblast will make it, but basement membrane is not made by the fibroblast. It's made by the epithelial cell. So that's it's part of epithelial tissue, right? It's not part of connective tissue. It belongs to the epithelial tissue. Endothelial cells are epithelial cells. They're squamous cells which are lining blood vessels. But here where the interesting feature comes in, because basement membrane of the glomerulus is not only made by the endothelial cells, it's also made by the podocytes. That's why it's extra thick. It's not normal thickness. 2 cells are making it, so it's extra thick. You can see it with pus. Remind me why? Why can this basement membrane can be seen with pus?
Glycoprotein? Collagen?
Amazing. Collagen is a glycoprotein. You remember that there are 437, right? So 437, these are the collagens in the basement membrane, and they are glycoproteins. And not only, as you remember, there are also some things, for example, multi-adhesive glycoproteins, proteoglycans, GAGs, right? So they're also very sugary, so pus will show you. So let's take a look. What do we have here? So this is a cross section of your glomerulus. Which cell is this? Knowing that this is blood.
endothelium.
Endothelial cell, very good. You can see it has some kind of holes. Which holes are these? Fenestra. How big are they?
70 to 90.
According to the lectures last year, 70 to 90 kilodaltons. According to the book, 60 kilodaltons. So I don't know. Check with the lectures this year. What is this?
Basement membrane.
Basement membrane, which was made by 2 cells, such as
Endothelial and podocytes.
podocyte. So you can see outside is the podocyte with so many processes, right? So there's so many processes there. The importance of the podocyte that it's also kind of part of the filtration barrier. So it's also participating in filtration because Between food processes of the podocytes, you can see that there are some kind of slits, and you call them filtration slits. According to the book, filtration slits are about 40 kilodaltons, so they're even smaller. So definitely no protein can pass through this barrier. No protein can do that because such small numbers, right? So filtration slits, I repeat, these are just spaces between food processes of the podocytes. So here you go, these are the podocytes, right? And you can see that there's so many food processes there, and the gaps between them are called filtration slits. We have said that basement membrane is made by two cells, by the podocytes and by the endothelial cells. So that's why it's extra thick. Because it is so thick, it can actually be divided into 3 layers. The inner layer of the basement membrane, which is closer to the endothelial cells, this is known as the lamina rara interna. Middle lamina densa to the podocyte, lamina rara externa, right? So there are 3 layers. So could you please tell me the entire filtration pathway? So if I'm being filtrated from the blood to the urinary space, right? What am I passing through? What is the first thing?
The endothelial fenestra.
Fenestra, very good. Endothelial fenestration, then?
The lamina rara.
Lamina rara interna, be more specific, then?
Lamina densa.
Densa.
Lamina rara externa.
And
then the podocytes.
filtration slit. Okay, so it's
Filtration
very
slits.
important because they will give you the option that says podocyte. You don't pass through the podocyte, you pass through the food processes slit. Okay, so the area where there is 2 food processes, so this is called the filtration slit, and you pass through this and you go to the urinary space.
This is so boring.
Are you serious? I actually like it because it actually makes sense. It actually makes sense. But wait, we haven't started the fun part. Okay, so yeah, now the fun part begins. So we have said that this is your glomerulus, okay, and it has 2 things, 2 cells. It has the endothelial cells and it has the podocytes, right? But now we're introducing the 3rd type of cells, which are called mesangium. Mesangium consists of 2 types of cells. They're intraglomerular mesangial cells and they're extraglomerular mesangial cells. So 2 more are going to be here, intraglomerular and extraglomerular mesangial cells. Of course, you can guess that which one is right next to the podocyte. Intra. Intraglomerular, because this is the glomerulus right here. So intraglomerular mesangial cell is going to be right next to the podocyte. Intraglomerular mesangial cell has these functions. So the most important being support and phagocytosis. Second most important, that it can contract. So it can actually kind of modify the size of your glomerulus, right? So intraglomerular mesangial cell. This one is not important. Next semester you will talk about it. So it can make some growth factors as well, but it's not important. We have said that there's also extraglomerular mesangial cell, right? Because so far we talked about intraglomerular mesangial cell, which I repeat is right next to the podocytes. It's part of the glomerulus. Phagocytosis things, supports things, etc. Where is extraglomerular mesangial cell? Well, obviously outside of the glomerulus, but where exactly? In a structure which is called juxtaglomerular, L is missing, juxtaglomerular apparatus. So extraglomerular mesangial cell is a part of this apparatus. Where is this apparatus? At the vascular pole. Do you guys remember vascular pole? Right, there is afferent, efferent arterioles, and here we have vascular pole. Right here is the juxtaglomerular apparatus. Juxtaglomerular apparatus consists of 3 types of cells. One of them is extraglomerular mesangial cell. Another ones are juxtaglomerular cells, and another one is macula densa. So 3 types of cells are making up this apparatus. What is the job for this apparatus? Why do I need it? Just one word, RAS. Okay, so this system, renin angiotensin-aldosterone system is activated thanks to juxtaglomerular apparatus. That's what it does. It activates the RAS system. So let's talk about macula densa cells, right? So the first cells of the juxtaglomerular apparatus, where are they? We have said this is a vascular pole because that's where the afferent and efferent arterioles are located. And we have also said that here we have this structure. Do you guys remember what was it called? Proximal was at the opposite end.
So distal convoluted.
Very good. So this one is the distal convoluted tubule, which is at the vascular pole. It's within the proximity, right? Proximal convoluted begins at the bottom of the urinary pole. So the cells of the distal convoluted tubule, which are very close to the vascular pole, they are known as the macula densa. So macula densa cells, I repeat, these are the cells of the distal convoluted tubule, but they are not present everywhere. Only the part which is touching the vascular pole, only that area, only there, the cells of the distal convoluted tubule are called macula densa. What is their job? Macula densa have osmoreceptors, so they could detect the concentration of some electrolytes, some ions. For example, sodium chloride. That's what macula densa cells are doing. They are sensing the concentration of the sodium chloride. What does it mean if I have very low sodium chloride?
And you're missing salt.
Basically, but what does it mean for my body?
Your blood volume is less.
My blood pressure is low. My blood pressure is low. It's really low. Salt is important in moderate amounts, okay, because it keeps your blood pressure normal. But if you have no salt, if you have no sodium chloride, your blood pressure is low. So what would macula densa want to do? Well, they would want to
Sense the sodium chloride.
yes, but if the blood pressure is low, what do they want to do? Okay, I sense that it's low. What do I want to do? I need to increase the blood pressure somehow, and one of the ways is definitely renin. I need to do something with the renin. So what do macula densa cells are doing? They are sending a signal to other cells nearby, to them here, and these cells are called juxtaglomerular cells. Juxtaglomerular cells are smooth muscle cells of the afferent arteriole. Okay, that's what they are. Juxtaglomerular cells are smooth muscle cells of afferent arteriole. Yet they make renin. So that's the part where your brain should be like, what the hell? Smooth muscle cell is making renin. Is juxtaglomerular cell an endocrine cell?
Yeah.
Yes, because it's making renin. So you're forgetting the part that it is smooth muscle cell, but because it is making renin, yes, it is an endocrine cell. So macula densa cells will be activating juxtaglomerular cells right here, and they'll make renin. And as you know, what will renin do?
From angiotensinogen to angiotensin 1.
Which will go and activate aldosterone, and sodium will be reabsorbed from the kidneys, water will follow, blood pressure is high. But what if it's urgent? What if you don't have time to wait until macula densa is sensing and then activating and then aldosterone formation? What if you don't have time? Can juxtaglomerular cell affect blood pressure directly? Can it do so by itself?
Contract or something?
Amazing. Don't forget that it's also a smooth muscle cell. So what will it do? It will constrict this vessel right here. It will contract. And if it contracts, is it easy for the blood to flow this way? No. So how's the pressure behind the afferent arteriole? It's super, super high. It's very difficult. So that's why juxtaglomerular cell, there was a question in the SCT, which is also asked in the oral. Juxtaglomerular cell basically is not waiting for the macula densa. So you don't need only stimulation from the macula densa to increase blood pressure. Juxtaglomerular cells are smooth muscle cells of afferent arteriole. Before macula densa senses that there is low sodium chloride, this smooth muscle cell already feels that the blood pressure is low because it's in the arteriole. It can tell you, is it stretched because the pressure is high, or is it not stretched, right? So it already feels by itself that it's like the blood pressure is not high. So it will constrict, make renin without macula densa. But of course, macula densa is important. So these were the two cells of the juxtaglomerular apparatus, right? So macula densa, juxtaglomerular, but don't forget there's the third one, extraglomerular mesangial cell. What does it do? Nobody knows. Probably makes erythropoietin. Probably. What's the exact function? Nobody knows. So here is your distal convoluted tubule. Here are the macula densa. This is your afferent arteriole. These cells are endothelial cells. Do not confuse them with the smooth muscle cells. These are endothelial cells of the blood vessel. Now these ones are your juxtaglomerular cell. Does it look like smooth muscle? Absolutely not. Okay, so as you remember, smooth muscle is very wavy and it has very elongated nucleus. So this kind of proves to you that they are actually endocrine cells, that these muscle cells are actually endocrine. So these ones are juxtaglomerular. And then these ones, which you can see here, for example, these are the extraglomerular mesangial cells. They have a bit darker nucleus. Okay, what does proximal convoluted tubule is doing? These things. So as you know, this is the first tubule to receive this urine from the urinary space, right? Bowman's space, Bowman's capsule. So it actively transports ions, reabsorbing water, glucose, amino acids, proteins, and vitamins. Whatever proteins managed, it will do so. To where? To where does it reabsorb?
To the capillaries, forget the name,
Yes.
tubular.
Peritubular capillary network. Okay, so don't forget proximal convoluted tubule will reabsorb not to the vasa recta, but peritubular capillary network. Another quite important thing is that the cells in the proximal convoluted tubule look something like this. This is the apical domain. This is the lateral domain. And this is the basal domain. And that's another lateral domain. So do you see? That's what the cells in the proximal convoluted tubule look like. The cells that are lining it. They have so many striations, right? So they have microvilli on one side. They have plica or folds on the lateral side, and they have basal striations at the basal side. So they have so many folds, so they can absorb as much as possible, because that's the first place where you're starting to absorb things, right? Loop of Henle, we talked about it. How do we call this? This is going to be the thick descending, or another name?
Straight, proximal straight.
Proximal straight. How do you call this one?
Distal straight.
Distal straight or thick ascending, and then just the thin components. What's the job of the loop of Henle? It has to generate high sodium concentration. That's the job. So you reabsorb other things as much as possible, keeping sodium high here. So sodium will not be reabsorbed here. Okay, so you want to keep it high here. How do you do that? Sodium, potassium, ATPase. The rest is not as important. Distal convoluted tubule. That's what you do here. You remove sodium and add potassium. So if in the loop of Henle you want to keep the sodium here, in the distal convoluted tubule, sodium should leave. And instead you add potassium. Why? Why would you rather excrete potassium and not sodium?
For your heart.
Amazing. What happens to your heart if there's too high potassium.
Good stuff.
It will. There's going to be prolonged hyperpolarization. The action potential will not be initiated whatsoever, and it will stop eventually. So that's why it's actually one of the easy death. Not so easy, it's very painful, but it's one of the easy deaths. All you have to do is inject potassium and the person will die. And if you live in the third world countries, they don't do well autopsy, so it will look like the person died from a heart attack. That's it. But of course, in the first world countries, they do autopsy, they look at the blood, and then they will notice that in your blood there is like so much potassium. So definitely you were killed, definitely. But yeah, it's actually one of those deaths in the nursing homes. Yes, I was just gonna say in the nursing homes, like they do it, actually. They just inject the potassium. Okay. You guys are so lucky. I'm not only teaching you history, I'm also teaching you how to kill people. That's insane, actually. Like, you know, it's insane, and it is on video. Okay, but yeah, okay, so yeah, but I fully trust you. But you guys are in medicine, so you shouldn't kill people, right? So that's what we're here for. We're actually trying to save them.
But we also live in Hungary, so.
True that. True that. Yeah. Okay, guys, so collecting system, that's the last part. So what do we do here? Very important, ADH works here. So antidiuretic hormone works on the collecting system. So could you tell me who makes ADH?
The adrenal
medulla? Yeah.
ADH, you're confusing.
No, it's the quart.
The liver.
You're confusing with aldosterone. ADH.
The liver.
No, not the liver. Well, I definitely did not expect it from this group, actually. Even my dumber groups have said it, but you guys, I was actually expecting that you'll say it immediately.
I'm very glad.
But it doesn't make it.
It's the pituitary. It's the.
It doesn't make it.
Hypothalamus.
Thank you more. Okay, so hypothalamus will be making the ADH, and posterior pituitary will be storing. In which area? How do you call the part where you store it?
Nervoso.
That's just okay. Fair enough.
The posterior.
Area, but how do you call the thing where you put it?
Bodies.
Herring's bodies, very good. The Herring's body. So ADH is stored in the Herring's body of the pars nervosa of the neurohypophysis. So as you remember, it is the antidiuretic. Another name is vasopressin, right? Because it increases your blood pressure. So ADH will be working on the collecting ducts. Your collecting ducts are going to express aquaporin 2 specifically. And these are the water channels. So all of the water will be leaving the collecting ducts and going into the blood. So it doesn't want you to pee out the blood, the water. Okay, so it doesn't want you to pee out the water. So antidiuretic. Okay, blood supply of the kidney, another very important one and very confusing one. You need to know this in the anatomy practice as well. So it's not enough to simply say that blood supply of the kidney is renal artery. It's not enough. You need to say all of those things. So let's begin. So here we have renal artery. It enters at the hilum, right, of the kidney. And in the hilum of the kidney, it divides into several branches. We call them segmental arteries, right? So typically they're about 2 to 3, 3 to 4, not more. So in the hilum of the kidney, it divides into the segmental arteries. Segmental arteries continue upwards, and you can see that they're running between these structures. So how would you call it?
Interlobular.
Interlobar. Yes, so you'll call it interlobar because this is a lobe, right? This is a lobe. So between the lobes, interlobar. Interlobar artery continues upwards. So for example, here, this is interlobar artery, continues upwards, and then it makes a 90-degree turn. And now it runs between cortex and medulla. So how would you call it here? This is called arcuate. Interlobular is here. Okay, so we'll talk about it. So it was my fault. So between the cortex and medulla, this area, this is known as the arcuate artery. So interlobar becomes arcuate. Arcuate is going to be kind of dividing. You will see it. What do I mean right now? Arcuate will give you a vessel down and a vessel up. A vessel up is called interlobular artery because you remember that here in the cortex we have lobules, right? So medullary rays, blah, blah, blah. A vessel down does not really have a name, but you would just kind of call it like a branch of the arcuate. It doesn't have a name, just a branch of the arcuate. Here in the medulla, this branch of the arcuate could become, for example,
Vasa recta.
yes, very good. It will become vasa recta. So it will become vasa recta. So, because you remember it's there, right? In the cortex, the interlobular that you see here, this interlobular will become afferent arteriole, which will become glomerulus, which will become efferent, and efferent will give you peritubular capillaries. So peritubular capillaries come from the efferent arteriole specifically. Okay, so directly. So peritubular capillaries, as you remember, they surround the tubules in the cortex, so they come from there. Vasa recta is debatable. So for example, I took it from the internet. So they said that vasa recta also comes from the efferent arteriole on this picture. But in our book, they say that vasa recta does not come from there. It comes as a branch of the arcuate. Okay, it comes as a branch of the arcuate because you're going down like this. Yes? What happens eventually? Eventually, this arcuate and this, this vasa recta and this peritubular capillaries will now drain to veins, some kind of branches. So eventually, through lots and lots of branches, they will end up in the interlobular, even vasa recta as well. So vasa recta will also kind of go upwards like this as well, okay? And it will eventually end up in the interlobular veins. From there, you go to arcuate, interlobar, segmental, and then renal. So I guess the most confusing part is this part. So what is happening here? So let's do it kind of again. So in the cortex, we have interlobular arteries. Interlobular artery is a branch of arcuate. From the interlobular, you have afferent arteriole, then glomerulus, then efferent, right? Because that's your glomerulus, that's your renal corpuscle. Afferent, glomerulus efferent. From the efferent, you have peritubular capillary network. Peritubular capillary network drains back into the interlobular vein. So in the cortex, it's very easy. Interlobular artery, afferent arteriole, glomerulus, efferent arteriole, peritubular capillary network, interlobular vein. In the medulla, it's a bit more difficult because here it's not a direct branch of anything. It's just like a result. So these vasa recta vessels, they in a way come from arcuate, right? So they come from arcuate vessels, but they drain eventually into the interlobular veins as well. So they don't go into the arcuate vein directly. They first go to the interlobular vein and then from the interlobular to the arcuate. So you make like additional stop.
But wait, they are going up, right? Because
Yes, vasa recta, yes, are going upwards, yes, but not to the cortex. So, well, in the medullary array, you will have them, yes, vasa recta. In the medullary array, you will have them there. Yes. Okay, that's the pathway for you guys. So podocytes, okay, filtration barrier, things that can pass are that cannot pass are here, things that can pass are here, right? So that can be filtrated. Notice majority of the time is just protein things, right? So proteins cannot go because they're too big, but also huge anions. Why? Because lamina rara interna, right, so the first part of the basement membrane, contains heparin sulfate. And as you remember, it's highly negative. So presence of heparin sulfate in lamina rara interna kind of repels large anions, so they don't want to go. Tips in the cortex, I'll explain it. So kidney, coronal section, capsule, cortex, medulla. Okay. How would you call this entire structure, knowing that this is cortex and this is medulla? How do you call this entire thing?
Lobe.
Lobe, very good. So this entire thing is called renal lobe because it's the medulla with the overlying cortex, yes. Inside of the cortex, you can see these dots. What are they?
Renal corpus.
Renal corpuscles. And you can see straight lines. In the cortex.
Medullary rays.
Yes, these are medullary rays, right? So you can see clearly in the cortex, there's some kind of straight protrusions, medullary rays. What is their other name? Pars radiata. Okay, so they're going to be known as the pars radiata. You can see between the cortex and the medulla, there's some kind of vessel here. You magnify to it, here is this vessel, and here's the artery to it. So vein and artery. How would you call them between cortex and medulla? So you remember that between cortex and medulla, what do we have?
Arcuate.
Arcuate. Okay, so what are these ones? These are also arcuate. So this is arcuate vein, and this is arcuate artery. A bit visible here, like a tiny lumen, but still there, like a branch. What about this one? As you can see, it's a bit on the side.
Interlobar.
Yeah. This is called interlobar because this is a lobe. So on the two sides of it, right? So this is an interlobar. This is minor calyx. And this is a renal sinus. Renal sinus is like an area where minor calyx becomes major. So it's just like a transitional point between minor and major. So that's what we see here. This is a minor calyx, and this area where adipose tissue is, this is a renal sinus. Okay, we have said that these are medullary rays, right? So pars radiata. What about this one?
Pars convoluta.
Convoluta, very good. These are the renal corpuscles. What about these vessels? Like these one, these one, these ones?
Intralobular.
Inter, right? So interlobular, yes. These are the interlobular vessels. So you can kind of draw imaginary line here because it's interlobular vein, right? Here we have some branches of this vein as well. So interlobular vein here as well. Draw a line. Whatever is present between these two imaginary lines, this is your renal lobule. So you notice that medullary ray is in the center of this renal lobule, surrounded by the cortical labyrinth. Okay, capsule, we can see that there is some kind of area here, which is called cortex corticis. It's never written, but is always asked. Cortex corticis is cortex of cortex. And it's very important because here you will not find any renal corpuscles. So that's your cortex corticis. And you will notice that no renal corpuscles are present there. All of them begin below this area. So let's do it again. What is this? Medullary ray. What is inside of it?
Loop of Henle.
Be more specific than loop of Henle. So you have to say
The thick.
Straight.
proximal straight, distal straight, and cortical collecting duct. Cortical collecting duct. What about here? This is pars convoluta. So what should be here?
Proximal and distal tubules.
proximal and distal convoluted tubules, and of course, renal corpuscles. Speaking of renal corpuscles, so let's take a look. What do we have here? Of course, in the center of it is glomerulus, right? So you can see blood vessels with the blood inside. There are some cells which are pale, and there are some cells which are dark. The paler cells are podocytes. Okay, so the pale cells, they are podocytes, like these ones with the pale nucleus. They are podocytes. The darker cells are intraglomerular mesangial cells. Okay, so intraglomerular mesangial cells, these ones. Do you guys remember what is the function for them?
Erythropoietin, maybe?
That was extraglomerular mesangial cell. But intraglomerular mesangial cells are simply
Structural.
structural support, phagocytosis, modulate the level of distension, like just they have contractile abilities, so etc. You can see there's some kind of squamous cells here, so
Endothelium.
so if these are the podocytes, on the other side,
The extra.
these are the parietal cells of the Bowman's capsule, right? So together, podocytes and the squamous cells, they're just the Bowman's capsule, visceral and parietal layer. So these ones are the parietal cells of Bowman's capsule. How do you call the space?
Urinary.
Urinary or Bowman space. And you can see that a structure begins at this area. So what is this?
Proximal convoluted.
Proximal convoluted tubule. So don't forget it always begins right from the urinary space. So this is a proximal convoluted tubule. If this is a proximal convoluted tubule, that means that this area right here is the urinary pole, right? It means that the opposite end is
The vascular.
pole, and you can clearly see how a vessel is coming in, right? So very nicely it's visible how a vessel is going upwards.
So that's an afferent?
Hmm? It is an afferent because of these cells.
The smooth muscle cells.
Yes, but they don't look like smooth muscle cells. So what are they?
They're endocytes.
These are the juxtaglomerular cells. Do not confuse them with these ones. These are endothelial cells. They're not smooth muscle cells, right? So these ones are endothelial cells. So these ones are juxtaglomerular cells. And what do they make?
Renin.
Renin. They're going to be making renin. Here you can see a structure.
The distal convoluted.
The
Yes,
distal
this is the distal convoluted tubule at the vascular pole. And you can see the cells here. These ones are going to be macula densa. Right, so what do macula densa do?
Tell the juxtacrine to make renin.
So they have osmoreceptors before that, right? So sense the sodium chloride. And if it is low, it's going to be activating juxtaglomerular cells. And last cells here outside of the glomerulus, which cells are these?
Extra.
Glomerular mesangial cells. So these ones are the extraglomerular mesangial cells, and maybe they are making erythropoietin, maybe. Okay, so how do I know that I'm looking at the distal convoluted tubule or proximal? Look at the color. The eosinophilic ones are proximal convoluted tubules. They are eosinophilic because they have lots and lots of transporters, and all of these transporters have a positive charge. So that's why you're going to see that eosin absolutely adores them. It will come and stain them nicely in the eosinophilic color. Distal convoluted tubule is a bit faint. Do you see? It's not as red. It's a bit faint in color. So the reason because it lacks these transporters. What about these structures between them?
Capillaries.
Yes. How would you call them?
Where are we? In the cortex or in the...
Peritubular capillary network, very good. So don't forget that you reabsorb into here, right? So proximal, distal, you reabsorb into here. So this is your peritubular capillary network. You can see endothelial cells of them everywhere. So these are endothelial cells. If we magnify, for example, here, this is a peritubular capillary network, right? So outside of them. Okay, everything else is just written there for you. Okay, so this is from the book. By the way, this is not our Ross, this is American books, because I felt like Ross kidney was horrible. So I had to open my American books and give you from there. Okay, so what do we have here? Weird looking slide. So how can I identify what am I looking at, right? So your job is to always identify this structure. What is it?
Renal corpuscle.
Renal corpuscle. If I see a renal corpuscle, I'm definitely in the cortex, not in the medulla. I'm in the cortex. So how do you call the area where the renal corpuscle is located? How would you call this? You will call it pars convoluta, because that's where the renal corpuscles are sitting, pars convoluta. If this is pars convolutum, that means that whatever tubules are outside of it are convoluted. So if I see a red tube outside of it, this is proximal convoluted. Why? Because it has more transporters. It's the first area where you're trying to reabsorb as much as possible. If I see a faint tube right next to it, this is distal convoluted tubule because it lacks said transporters. And then you see a structure right next to it, which also has red tubes and faint tubes with a lumen or without a lumen, but they are not convoluted. They are straight. So how would you call this area then?
The medullary ray.
Medullary ray, or another name, pars radiata, because it is straight. So you remember that within the medullary ray, we have proximal straight, distal straight, or cortical collecting duct. The easiest to identify is a cortical collecting duct. Why? It's the biggest there is. So if you see a lumen and you see faint color, this is a cortical collecting duct. If we're talking about medullary ray specifically, right? So this is medullary ray right next to my pars convoluta. So this is a cortical collecting duct. Now you have two ducts left. One of them is red. One of them is eosinophilic, no color, but without lumen. You cannot see the lumen. The lumen is very condensed. It's very packed. So what do you think? If it is red, then it is
Proximal straight.
very good. Proximal straight, very similar to your part here, right? So this is proximal straight. If it is no color, faint, distal straight. Your turn. So how do you call this?
Pars
Pars convoluta. How do you know?
because there's renal corpuscles.
Exactly. Guys, sometimes some professors will not say pars convoluta because it is in Latin. Sometimes they will say cortical labyrinth. It's the same thing. So pars convoluta or cortical labyrinth because it's very labyrinthic. I don't know. So yeah, so this is going to be the cortical labyrinth or pars convoluta, the same thing. Right next to it, with this structure included,
Medullary ray.
medullary ray. And then again,
Pars convoluta.
pars convoluta. And then again,
Medullary.
medullary. And you see they're changing. I see a vessel here and I see a vessel here. So I draw imaginary line like this. So how do you call it between the two lines?
Renal lobule.
Renal lobule. So let's take a look. We're looking at the medullary ray. The easiest to identify are huge structures with huge visible lumen, which have no color. So how do you call them?
Cortical.
Cortical collecting ducts. Second easiest, huge, eosinophilic.
Proximal straight.
Proximal straight. And then if I see something like this, or like here, look how many there are, okay, or like this one,
Distal.
distal straight. So almost no lumen and very no color, very no color. Okay, so yeah. How would you call this one?
Proximal convoluted.
Proximal convoluted, very good. How would you call this one? Or this one? Or this one?
Distal convoluted.
Distal convoluted. Is it clear?
Yes.
This is a kidney as well, but I rotated it, different section, and I have not put it into the physiological appearance. I completely rotated it. So tell me, how would you call this?
Sparse convoluted.
It's too specific. Generally,
Cortex.
just cortex. Why?
Because the renal corpuscles.
You see
Exactly, because renal corpuscles are here. How would you call this one?
the medulla.
Medulla, because there aren't any. So that means that which vessel is this?
Arcuate.
Arcuate artery. That means that which vessels are these?
Their loba.
Ether
Lobular.
lobular. They're in the cortex, right? So they're within the cortex. So yeah. Okay. To which nephron does this renal corpuscle belong?
Superficial.
Superficial cortical. So where is its loop of Henle? Barely touching the medulla, barely, like this. To which nephron does this renal corpuscle belong?
Juxtaplant.
Juxtamedullary. You can see it's adjacent to the medulla. So where is its loop of Henle? Here. All of them here, these are going to be loop of Henle. Okay, minor calyx we talked about, renal sinus we talked about. You remember that renal sinus is just the area that is between the minor and the major calyx. So the epithelium here is going to be ura epithelium. We will talk more details about it next class. Okay, now we're going to go to the medulla. And that's the part that confuses everyone. So now we're in the medulla. So immediately from this slide alone, how do you know that you're looking at medulla?
Now renal corpuscle.
Exactly, you cannot see renal corpuscles. So remember that there aren't any renal corpuscles in the medulla. So what should you see in the medulla?
The ducts.
Loop of Henle.
Loop of Henle. Don't forget, loop of Henle has 4 parts. So which part should be seen there?
The medulla. The thin.
Thin only? Can there be thick?
Yes, if it's a juxtamedullary.
Amazing. If it is juxtamedullary, you can also see thick. So this is juxtamedullary. So these thick components could also be seen in the medulla. So basically the entire loop of Henle could be seen, both proximal and distal straight, including the thin ascending, descending as well. What else can be seen in the medulla?
Medullary collecting duct.
Very good. And lastly, what else could you see there?
Papillary.
Recta.
Papillary is huge. You can see it with a naked eye. On the histo, you will not see it, but in anatomy, you will.
Vasa recta.
Vasa recta. The easiest one for me to identify personally are vasa recta. If I see very tiny structures with a blood in it, this is vasa recta. Okay, so yellow, these are vasa recta. Second most easiest for me to identify are collecting ducts, medullary collecting duct. I find the biggest there is because collecting ducts are generally very big, a huge lumen, faint color. So in black color, these are my medullary collecting ducts, right? So medullary collecting ducts are these ones.
What are the cells around them?
These cells, these are the lining cells of the collecting duct. So you know the duct consists of cells and basal membrane, right? So these are just that part. So these cells are the same cells which we see here. these ones, just different section. So here it is longitudinal section, there it is a cross section, right? So it is a cross section of that duct. Next one are the distal straight. They are a bit similar to the size of collecting duct, just a bit smaller, and the color is faint, right? So the purple, these are the distal straight. Looking like distal straight, but a bit red, right? These ones, they are the proximal straight. And everything else, super tiny, which you can see here, right? Super tiny, very, very thin. These are the thin segments of the loop of Henle. And notice how many there are. There's so many of them. Yes?
Which one is the thin?
Everything. So blue color, like light blue, these ones, these are the thin segments of loop of Henle. This one as well. This one, there's so many, these ones, all of them, the rest of them basically. Everything that is not is a loop of Henle. A thin segment of loop of Henle.
Okay.
And last thing for today, we have a tangential section. Didn't know how to explain it, so here we go. Okay, it's like a periphery section. I don't know. You have an organ and you longitudinally cut it peripherally. Because longitudinal section is like this, it's like almost in the middle, but tangential is like you're cutting periphery. What's the purpose of tangential section? To show cross section of medullary ray. Because so far we've seen it longitudinal section, but this section is needed to show you cross section. So you start with something like this. So green part, this is the medullary ray. Blue, this is the pars convoluta, right? So this is the pars convoluta. So everything what is within the blue color, this is cortical lobule, but a better said, this is renal lobule, remember? So renal lobule. So how do you call a vessel which is at the border of it?
Interlocutor.
Interlobular. So here this is interlobular vein and this is interlobular artery. Okay, so two vessels are visible. So these are them. So we look at this. Everything is a mess. Do not understand what am I looking at whatsoever. As always, nothing has changed. Your job is to identify the structure. So renal corpuscle is visible. That means that whatever is surrounding it is the cortical labyrinth or pars convoluta, right? So everything which is within the blue borders, this is the pars convoluta because that's where the renal corpuscle is. So knowing that this is pars convoluta, what is this?
Proximal convoluted,
Proximal convoluted, and this one is
distal convoluted.
distal convoluted. And then you look at the periphery of the structure. Do you see any renal corpuscles here?
No.
No. So it cannot be pars convoluta. It has to be
Medullary.
pars radiata or medullary rays. And that's exactly what it is. You see that at the two structures here, these are the medullary rays, like parts of the medullary rays. So how can you identify what is what? So what could this be if it is in the medullary ray? The biggest ones.
Collecting that.
Cortical collecting ducts. The smaller ones.
This, it's not thick.
Distal straight. The red ones,
Proximal.
proximal straight. Let's do it again. So what are the bigger ones? We are still in the medullary ray. Notice there is no renal corpuscle whatsoever within the proximity, right? So what are the green ones?
Cortical collecting duct.
Very good. These ones,
Distal.
distal straight. These ones,
Also distal.
look at the color. It's a bit different.
Ah.
Here, the nucleus is
proximal.
more, right? And the color is different. So these ones are the proximal straight. So notice the difference. This one,
Distal straight.
Distal.
are you sure? Look at the color.
Proximal.
It's very red. The color is very eosinophilic. I know it looks like one right now, but notice that the nucleus is not as prominent. So it's a proximal straight. Right next to it,
That would be
Distal.
distal straight. What about this one? A blood vessel between them.
Peri.
Vasa recta.
Vasa recta. Vasa recta. Why vasa recta?
Lines in the
Because this is
In
loop
the
of Henle.
medulla.
The proximal and the distal straight, they are still parts of the loop of Henle. So blood vessel between them is not peritubular, it's a vasa recta. This one?
Collecting. No.
Acute.
It's not blood. Because look, red blood cells are looking like this and they're not here.
Proximal
Sorry?
collecting duct.
We don't have proximal collectin. We have a cortical collectin or medullary collectin, but we have two proximal, either straight or
Convoluted.
convoluted. So this one is actual proximal convoluted duct. So even though you do not see glomerulus nearby, I mean, there is one here, but still notice the difference. So this is definitely proximal straight, and proximal convoluted is a bit bigger, right? What about this one?
Distal convoluted.
Distal convoluted. And how do you call this part between them?
Very.
Peritubular capillary network, yes.
How are the tubules bigger than the collecting ducts?
Good question, but these are cortical collecting ducts, so they are just starting. They're just starting, so they're not huge immediately because you're just starting to collect the urine. Now, medullary will be the size like this, yes. Medullary will be quite big, yeah. And that's it. That's where I want to finish. So, what should you do? You should study this for the next class because I will be asking you.